Hello everyone, this is Dr. P. Mary Anupama, Assistant Professor from the Department of Biochemistry, St. Joseph's College for Women, Visakhapatnam. Today, let us study about nucleotide metabolism. The contents of this presentation include sources of the atoms in the purines and pyrimidine molecules, biosynthesis and degradation of purines and pyrimidines, regulation of purine and pyrimidine synthesis, and inborn errors of nucleotide metabolism. First, let us see the sources of atoms for purines. Many compounds they contribute to the purine ring of nucleotides. So here we'll start with N1, C2, N3, C4, C5 and C6, followed by N7, C8 and N9. We shall see what groups contribute to these atoms. N1 of purine is derived from the amino group of aspartate. C2 and C8, these arise from the formate of N10 formyl tetrahydrofolate. N3 and N9 they are obtained from amide group of glutamine. C4, C5 and N7 they are contributed by glycine, and C6 directly comes from carbon dioxide. Now let us start with purine biosynthesis. Purines, these are built upon a pre-existing ribose 5-phosphate. Liver is the major site for purine nucleotide synthesis. Erythrocytes, polymorphonuclear leukocytes, and brain, these are the three which cannot produce purines. And the pathway for the synthesis of Inosin Monophosphate, which is IMP or inosinic acid, it consists of 10 steps, and we shall see this de novo pathway of purine biosynthesis. Now let us see the 10 steps of purine synthesis. The first step is synthesis of phosphoribosyl pyrophosphate. The ribose 5-phosphate which is produced from the HMP shunt, that is the starting material for purine nucleotide synthesis. This reacts with ATP, the ATP donates 2 phosphates, that is the pyrophosphate, which gets incorporated at the C1, resulting in the formation of 5-phosphoribosyl 1-pyrophosphate, which is the PRPP. This reaction is catalyzed by PRPP synthetase. The next step is the transfer of nitrogen from glutamine. Glutamine transfers its amide nitrogen to PRPP to replace the pyrophosphate to produce 5-phosphoribosyl amine. The enzyme which is involved in this catalysis is PRPP glutamyl amidotransferase, and this is controlled by feedback inhibition of the nucleotides IMP, AMP and GMP. This step, we call it as the committed step in purine nucleotide synthesis. And here the glutamine, after donating the nitrogen, is converted to glutamate, and then pyrophosphate is released. In the third step, phosphoribosylamine reacts with glycine in the presence of ATP to form glycinamide ribosyl 5-phosphate or glycinamide ribonucleotide, abbreviated as GAR. The next step involves N10 formyl tetrahydrofolate. The N10 formyl tetrahydrofolate donates the formyl group, and the product thus formed is formyl glycinamide ribosyl 5-phosphate or formyl glycinamide ribonucleotide. The next step is the transfer of again amino group from glutamine. Glutamine transfers the second amino group to produce formyl glycinamide ribosyl 5 phosphate. Here again ATP is required for this reaction. The sixth step involves formation of imidazole ring by the closure here. So the imidazole ring of the purine is closed in the presence of ATP. This is a ATP driven reaction, and the formyl glycinamidine ribonucleotide FGAM that gets converted to 5-amino-imidazole ribonucleotide AIR, or it's also called as 5-amino-imidazole ribosyl 5-phosphate. The next step is carboxylation reaction, that is incorporation of carbon dioxide. So the carboxylation, it yields amino-imidazole carboxylated ribosyl 5-phosphate. This we call it as carboxyl amino-imidazole ribonucleotide. So here we have the carbon dioxide which is incorporated, and this reaction, it is catalyzed by a carboxylase. The eighth step involves condensation of aspartate with 5-amino-imidazole carboxylate ribosyl 5-phosphate, and this results in the formation of 5-amino-imidazole 4-succinyl carboxamide ribosyl 5-phosphate, and this is again a ATP driven reaction catalyzed by a synthetase. The last step involves cyclization at this state, resulting in Inosin Monophosphate. This reaction is catalyzed by Cyclohydrolase. So with this we get this Inosin Monophosphate, and we have the purine synthesized. Synthesis of AMP and GMP from IMP. Inosin monophosphate is the precursor for the formation of AMP and GMP. Aspartate, it condenses with the IMP in the presence of GTP to produce adenyl succinate, which on cleavage forms AMP. For synthesis of GMP, IMP undergoes NAD plus dependent dehydrogenation to form xanthosin monophosphate. Glutamine then transfers the amide nitrogen to xanthosin monophosphate to produce GMP. 6-mercaptopurine is an inhibitor for the synthesis of both AMP as well as GMP. It acts on the enzyme adenyl succinase of AMP pathway and IMP dehydrogenase of GMP pathway. So here is the pathway from IMP. First let us see the synthesis of AMP. Aspartate, it contributes the amino group. So aspartate in the presence of GTP condenses with IMP. Now fumarate is eliminated from here, resulting in AMP or adenylate. This is a GTP dependent reaction. Now coming to the synthesis of GMP. Here initially IMP is converted into xanthosin monophosphate. This is a dehydrogenation reaction which is NAD plus dependent. IMP dehydrogenase catalyzes the reaction, and then glutamine is the donor for the nitrogen, and finally resulting in GMP. Once all the monophosphates are formed, that is purine monophosphates, from them the purine nucleoside diphosphates and triphosphates are to be produced. So the nucleoside monophosphates, that is AMP and GMP, are to be converted into corresponding di and tri through some metabolic reactions which are represented like this. So the nucleoside monophosphates AMP and GMP are initially phosphorylated to result in ADP and GDP by nucleoside monophosphate kinases. Now these nucleoside diphosphates are then phosphorylated further by ATP by the enzyme NDP kinases, that is nucleoside diphosphate kinases. Thereby we get ATP and GTP. Now let us talk about salvage pathway for purine synthesis. The free purines like the adenine, guanine and hypoxanthine, these are also formed during the normal turnover of nucleic acids, mainly the RNA, and these are also obtained from our dietary sources. So the purines, they can be directly converted to the corresponding nucleotides, and such process we call it as the salvage pathway. Adenine phosphoribosyl transferase that catalyzes the formation of AMP from adenine. Then we also have hypoxanthine guanine phosphoribosyl transferase, abbreviated as HGPRT. This converts guanine and hypoxanthine respectively into GMP and IMP. The phosphoribosyl pyrophosphate, which is abbreviated as PRPP, that becomes the donor of ribose 5-phosphate in this salvage pathway. So the adenine, which is obtained from dietary sources or from RNA, that is transferred with ribose 5-phosphate resulting in AMP, Adenosine Monophosphate. PRPP becomes the donor for ribose 5-phosphate and the reaction is catalyzed by adenine phosphoribosyl transferase. Same is the case with guanine. PRPP becomes the donor for ribose 5-phosphate, but the enzyme that catalyzes the reaction is hypoxanthine guanine phosphoribosyl transferase. Then when it is hypoxanthine, same thing, we get IMP after the transfer of ribose 5-phosphate by PRPP, and the reaction is catalyzed by hypoxanthine guanine phosphoribosyl transferase. The next component that we have to study is formation of deoxyribonucleotides from ribonucleotides. We have the ribonucleotide diphosphates which are produced, that is ADP, GTP, CDP, UDP, all these things are produced. Now these are reduced by ribonucleotide reductases and we will be getting deoxyribonucleoside diphosphates. This reaction requires the cofactor thioredoxin which contributes the reducing groups, and finally thioredoxin gets oxidized and we get deoxyribonucleoside here. This thioredoxin can be again obtained by thioredoxin reductase which requires NADPH as which is the donor for the reducing groups. So thioredoxin which is oxidized gets back to thioredoxin reduced form, and finally it is ready for another series of conversion of this ribonucleosides to deoxyribonucleotides or ribonucleotides to deoxyribonucleotides and so on. Now let us study about degradation of purines. The nucleotide monophosphates, that is AMP, IMP and GMP, these are converted to nucleoside forms, that is the adenosine, inosine and guanosine by the action of nucleotidases. The amino group either from AMP or adenosine, it can be removed to produce IMP or inosine respectively. The inosine and guanosine, these are converted to hypoxanthine and guanine by purine nucleoside phosphorylase. Adenosine is not degraded by this enzyme, hence again that has to be converted to inosine. And guanine, it undergoes deamination by guanase to form xanthine. That is what we'll study here. AMP, this loses its phosphate to produce adenosine, which again undergoes deamination to result in inosin. Or AMP, initially it may undergo deamination by AMP deaminase followed by remolar phosphate by nucleotidase resulting in inosin. And GMP, it loses its phosphate initially resulting in guanosin. Now this guanosin, it again undergoes dephosphorylation, that is it will be losing its phosphate, ribose 5 phosphate, and then you'll get guanine. So this part is lost upon phosphorylation of this ribose 1 phosphate releasing guanine. And here the inosin, it is again, and it undergoes the same reaction resulting initially in hypoxanthin. Then by the action of xanthin oxidase, it is converted to xanthin. So we have xanthin and guanine now. So this xanthin oxidase is an important enzyme that converts hypoxanthin to xanthin and then the xanthin to uric acid. The enzyme, it contains FAD, molybdenum and iron and is exclusively found in the liver and small intestines. Xanthin oxidase, it liberates hydrogen peroxide which is harmful to the tissues. So catalase, which is present here, cleaves this hydrogen peroxide into water and oxygen. The uric acid which is produced, it is the final excretory product of purine metabolism. And you can see even this guanine by the action of guanase is converted to xanthin. Then xanthin by xanthin oxidase is converted to uric acid. Finally, we have uric acid as the end product of purine metabolism. Now let us see the regulation of purine synthesis. The purine nucleotide synthesis is very well coordinated to meet the cellular demands. The intracellular concentration of PRPP, it regulates the purine synthesis to a larger extent, and this in turn is dependent upon the availability of ribose 5 phosphate and the enzyme PRPP synthetase. So if these are present, then purine synthesis will be taking place. The PRPP glutamyl amidinotransferase, it is controlled by feedback mechanism of purine nucleotides. That is, if AMP and GMP are available in adequate amounts to meet the cellular requirements, their synthesis is turned off at amidinotransferase reaction. Another important stage of regulation is in the conversion of IMP to AMP and GMP. The AMP, it inhibits adenyl succinate synthetase while GMP inhibits IMP dehydrogenase. Thus AMP and GMP control their respective synthesis from IMP by feedback mechanism. Disorders of purine metabolism. Uric acid, it is the end product of purine metabolism and its concentration in the serum of adults is in the range of 3 to 7 milligrams per deciliter of blood. In women, it is slightly lower by just 1 milligram than in men, and the daily excretion of uric acid is about 500 to 700 milligrams. Hyperuricemia, it refers to the elevation in the serum uric acid levels, and this is sometimes associated with increased uric acid excretion through urine, which is called as uricosuriia. One disease stage which is very prominent is the gout. It is a metabolic disease associated with overproduction of uric acid. At the physiological pH, the uric acid is found in more soluble form as sodium urate. In severe hyperuricemia, crystals of this sodium urate get deposited in the soft tissues, particularly in the joints. Such deposits, these are known as the tophi. This causes inflammation in the joints resulting in painful gouty arthritis. Sodium urate and or uric acid may also precipitate in the kidneys and ureters, thus resulting in renal damage and stone formation. Gout was found to be more associated with high living, overeating and alcohol consumption. The possible reasons for gout are the decrease in glucose 6 phosphatase, or it can be an increase in glutathione reductase, or an increase in PRPP synthetase, which can lead to increased synthesis of the nucleotides, or an increase in PRPP glutamyl aminotransferase, a decrease in HGPRT that leads to accumulation of the bases, nitrogen bases. So all these reasons can contribute to gout. The next part that we need to study is biosynthesis of pyrimidine nucleotides. Pyrimidine ring is firstly synthesized and then attached to ribose 5-phosphate, which is unlike the purine nucleotides. So here purine ring is first built and then it is attached to ribose 5-phosphate, and this is the pyrimidine nucleus, and you can see the contributors for various atoms. So N1, C6, C5 and C4, they come from aspartate, while N3 comes from glutamine and C2 comes from carbon dioxide. These are the core atom contributors for a pyrimidine ring. The first step is condensation between carbon dioxide and glutamine. Glutamine, it transfers its amido nitrogen to carbon dioxide to produce carbamyl phosphate. This reaction is ATP dependent and is catalyzed by cytosomal enzyme carbamyl phosphate synthetase 2, abbreviated as CPS2. The carbamyl phosphate now condenses with aspartate to form carbamyl aspartate, and this reaction it is catalyzed by aspartate transcarbamylase. The phosphate is eliminated. Next thing is production of dihydroorotate. This reaction is catalyzed by dihydroorotate and it is a ring closure reaction resulting in dihydroorotate. The pyrimidine ring closure takes place resulting in the loss of a water molecule. And the next step in pyrimidine synthesis is an NAD plus dependent dehydrogenation reaction that leads to the formation of orotate, and the reaction is catalyzed by dihydroorotate dehydrogenase. Now the orotate is transferred to ribose 5-phosphate, and the ribose 5-phosphate contributor is PRPP, that is phosphoribosyl pyrophosphate, and the reaction is catalyzed by orotate phosphoribosyl transferase and will be getting Inosin Monophosphate OMP. The decarboxylation of OMP that results in UMP, that is uridine monophosphate, and the reaction is catalyzed by OMP decarboxylase. Now here also we have UDP to DUDP production. So here ribonucleotide reductase, as we have seen previously, it converts UDP to DUDP, and the reaction is catalyzed by enzyme that requires thioredoxin. And UDP, it undergoes kinases reaction, that is phosphorylation reaction by an ATP dependent kinase resulting in UTP. So CTP is produced from UTP by elimination of UTP. Then CTP synthetase, that is the enzyme, and glutamine is the provider for the nitrogen during this elimination reaction. So here we can see UMP, which is produced. It is phosphorylated by a kinase, ATP dependent kinase, resulting in UDP. Now this UDP, it is the precursor for DTMP as well as the CTP. So UDP, it undergoes a reduction reaction, that is ribonucleotide reductase, that results in the formation of deoxyuridine diphosphate from UDP to DUDP. From that you get DUMP, from which you get DTMP. So here we have thymidylate synthetase, which takes up this methyl group, and finally you get DTMP. While here the DUDP, after it is converted to UTP by a kinase, it is again eliminated, resulting in CTP formation by the enzyme CTP synthetase. Next, talking about degradation of pyrimidines. The pyrimidine nucleotides, they undergo similar reactions like the dephosphorylation, deamination, cleavage of the glycosidic bond, just like our purine nucleotides, and they liberate the nitrogenous base, that is the cytosine, uracil and thymine. These bases, they are then degraded to highly solubilized forms or solubilized products like the beta-alanine and beta-amino isobutyrate. So these are the amino acids which undergo transamination and other reactions to finally produce acetyl-CoA and succinyl-CoA. Coming to the salvage pathway, pyrimidines, just like purines, they also serve as precursors in the salvage pathway to be converted to respective nucleotides, and these reactions, they are catalyzed by pyrimidine phosphoribosyl transferases, which then utilize the PRPP as the source for ribose 5 phosphates. So these transferases, they transfer the ribose 5 phosphate onto the pyrimidines if they are available either through the diet or during some degradation of nucleic acids. Regulation of pyrimidine synthesis. In bacteria, we have aspartate transcarbamylase, ACTase, which catalyzes a committed step in the pyrimidine biosynthesis. This ACTase is a very good example of an enzyme that exhibits this feedback mechanism, wherein the CTP, which is the end product, immediately stops if this is in excess, the first committed reaction. In certain bacteria, UTP, it also inhibits the ACTase. So, ATP, however, it is known to stimulate the ATCase activity, that is aspartate transcarbamylase activity. Disorders of pyrimidine metabolism. Auritic aciduria. This is a rare metabolic disorder characterized by the excretion of uric acid in urine, severe anemia, and retarded growth. It is due to the deficiency of the enzyme orotate phosphoribosyl transferase and OMP decarboxylase of pyrimidine synthesis. Both these enzyme activities are present on a single protein as domain. Feeding diet rich in uridine or cytidine is an effective treatment for uric aciduria. These compounds, they provide pyrimidine nucleotides required for DNA and RNA synthesis. Besides this UTP, it inhibits carbamyl phosphate synthetase 2, the first enzyme, and blocks synthesis of uric acid. Then we have Reyes syndrome. This is considered as a secondary uric aciduria. It is believed that a defect in ornithine transcarbamylase of urea cycle, there the carbamyl phosphate is produced. So this leads to accumulation of carbamyl phosphate in the urea cycle that is diverted here, and increased synthesis of uric acid will be taking place, and thereby you see uric acid in the urine. Thank you for watching this video. Please subscribe for further videos.